So for 40 plus years, I've enjoyed a career as a venture capitalist. When I started in the 1960s, nobody knew what the term meant. It was a cottage industry, and literally less than $100 million was raised each year and invested in private companies. Today, 5 decades later, venture capital is a respected asset class. Approximately $20 billion annually is raised and, in an efficient Darwinian manner, invested in the most promising high-tech startups and private growth companies. I've seen the power when you marry a visionary entrepreneur with Tier 1 venture capital financing. It's created companies that have changed your life and mine, like Apple and Facebook and Groupon. And it's created whole new industries, such as the biotechnology industry. 10 years ago, when she was 58 years old, my wife was diagnosed with Alzheimer's disease. And frankly, I was appalled when we went to the best doctors in Boston and we were told, try 3 Advil a day. It might help. Try Lipitor, an FDA-approved drug for lowering cholesterol. It might help. And oh yes, there are a couple of Alzheimer's disease-approved drugs like Reminol and Aricept. They won't help. This, for a disease that was discovered over 100 years ago, affects 5.5 million Americans today, and if we don't find a cure, will affect 16 million by 2050. Look to the person on your left and on your right. The statistics tell us that when the 3 of you reach 80, 1 of you will have Alzheimer's disease. So being a venture capitalist and being a curious guy, I've studied why we haven't made more progress in finding a cure. It is not for lack of cutting-edge, world-class researchers. They exist in our leading universities and our teaching hospitals throughout this country. But unfortunately, in my opinion, the financial model for funding a lot of medical research is severely challenged. It's insufficient, it's inefficient, and it's ineffective. It's insufficient when we spend $1 on Alzheimer's research for every $400 we spend on caregiving. It's inefficient when the heads of some of the best academic research laboratories in our country spend literally 30 to 40% of their day filling out bureaucratic forms in search of funding. And they don't hear back from their funding source for over a year, and even if they're successful, they don't get funded and see the money for over 2 years. And worst of all, it's ineffective because the research granting mechanism of so many well-meaning organizations has become so risk-averse that the projects they do fund advance the ball 1 yard at a time, or if you will, give a bond-like return. But fortunately, there is a new movement that's taken hold in this country in the last 10 years that uses venture capital principles to accelerate medical research. In my opinion, one of the founders was Mike Milken, who you just heard about, and all of his pioneering work in the last 2 decades on prostate cancer. And he continues his efforts with our last speaker's organization, Faster Cures. And there's a number of new disease-specific nonprofits that have been formed in the last 10 years using the venture capital approach, including Cure Alzheimer's Fund, which I co-founded 5 years ago. The venture capital approach is simple, it's results-oriented, and to me, it's common sense. First, we proactively identify world-class researchers, in our case in Alzheimer's disease, and we fund those people. Second, we eliminate the bureaucracy. They submit proposals to us on 2 pages, and we respond in weeks, not months or years. Third, we challenge these researchers to dare to be great, to do projects that, yes, have some risk, but if successful, will advance the ball 20 or 30 yards down the field, give you a venture capital-like return. And fourth, we insist that these researchers cooperate as a virtual dream team, sharing their data and their research collaboratively and transparently. I believe that if this venture research movement grows in the next 20 years, like the for-profit venture capital industry has grown in the last 20, that at a future Imagine Solutions conference, we'll be talking about a real cure for Alzheimer's disease. And now let me introduce Dr. Rudy Tanzi, a world-class geneticist at Mass General Hospital, to update you on Alzheimer's and to share with you how venture research funding has helped accelerate his race to a cure.
Thank you, Henry, for the introduction and especially for making all my research I'm about to tell this audience about possible. And good afternoon to you all. I'd like to start with 4 numbers. Every 72 seconds in this country, there's a new diagnosis of Alzheimer's disease. Over 85 years old, 40% of the population has Alzheimer's disease. With 72 million baby boomers heading toward risk age, we calculate that by 2015, the tipping point will be reached where Alzheimer's could potentially single-handedly collapse Medicare and Medicaid. It's a very expensive disease, as you just heard from Henry. And 0 corresponds to the number of drugs we have that can stop the disease and actually treat the disease at its roots. So I'm going to talk to you about a plan that I will stand by where we have the intention to eradicate, not cure, but eradicate Alzheimer's by 2020. And how will we do that? Well, first of all, venture research funding, venture philanthropy makes this possible, because what we're doing is a bit out of the box, and it's very difficult, as you heard, to get enough federal funding to do what I'm about to tell you in terms of what we've done over the last 5 or 6 years. The first clue comes from this. Everything we know, just about everything we know about Alzheimer's disease and its causes has come from genetic research. And our hope for eradicating this disease has this mantra: early prediction of the disease using genetics with proper legal protection, early detection pre-symptomatically for those who are at risk, and most importantly, using genes to teach us how to treat this disease at the level of prevention. Know you're at risk, detect early, prevent the disease before it strikes. So this is a pharmacogenetic approach similar to some of the talks you heard earlier in the day. Now, since Alzheimer first described this disease in 1906, for decades, we knew nothing about the cause. We just could explain the pathology: plaques, tangles in the brain was shrinking. So trying to understand the disease was akin to going to Foxborough Stadium at 5 o'clock after the New England Patriots played at 1 o'clock and trying to figure out the game of football. You just see basically the aftermath. And we were stuck there for decades. And it wasn't until 1987 when, taking advantage of the fact this is a very genetic disease, inheritance plays a huge role, that we and others were able to find 4 different genes. My lab discovered the first gene, APP, and we also co-discovered the presenilin genes, PS1, PSEN1 and 2, and ApoE is a gene that was discovered at Duke. But these 4 genes, for the first time, taught us what was going on in the disease, and let me show you what we learned. So, how do we take genes, and how do we get to therapies? You start with genes, and genes teach you about the proteins they make. A defective gene makes an abnormal protein. An abnormal protein doesn't operate correctly in some biological system or pathway. That gives you the target. Then you have to have the choke point. You have to say, here's what we have to go in and fix. Here's what's broken. Here's what we fix to treat the disease. And this is done with drugs or other therapies. Now, let me give you an example of that. Heart disease, okay? Many of you may not know that the reason why incidence for heart disease has gone down so much because of the use of statins is that Brown and Goldstein found by genetics that the cholesterol receptor had mutations in it, and these patients got very early heart disease and died. Based on that, they came up with a target: high cholesterol. So, and we all know what high cholesterol does for heart disease. So you look at the cholesterol pathway, how cholesterol is made, and basically what does a statin like Lipitor do? It hits that very first biochemical event by which cholesterol is made, and in this way, using just enough of the drug, you can bring your cholesterol down to safe levels. I'm telling you today that to prevent Alzheimer's in those at risk, we need the equivalent of a statin for Alzheimer's disease. How do we get there? And now we get back to genetics. All 4 genes point to the same thing. Our cholesterol for Alzheimer's disease is a little protein called A-beta. A-beta is made in the brain normally. You make it, you clear it. The brain's very careful not to make it accumulate too much, because if you get too much of it in the brain, it forms the senile plaques. But more importantly, little clumps of it get in the synapses where neurons are communicating and effectively cause a short circuit. It actually impairs neurotransmission if you have too much of it. So this has become the target. Now, where's A-beta come from? Well, this was the first gene we found. It makes this protein that I named amyloid precursor protein because it was the precursor of the amyloid. A-beta is also called amyloid beta protein. And the A-beta is that little red square in that larger protein in green. So what has to happen is you have enzymes, molecular scissors, that snip that little red segment out of the big green bar. And now you have made A-beta. The first 3 genes we found, APP is the substrate, it gets clipped. The presenilin genes we found in 95 do the clipping. And then A-beta gets made, and ApoE, the 4th gene, helps the A-beta to get out of the brain so you don't accumulate too much. So that's why I'm saying all 4 genes are all revolve around this A-beta in the brain. Now, what we know is that in Alzheimer's disease, you get too much of the A-beta accumulating. It starts to aggregate. When A-beta starts to aggregate into clumps, the brain has a much more difficult time getting rid of it. And now you start forming these plaques. And then up top, you see where the A-beta is getting in and impairing the neurotransmission, preventing learning and memory by actually getting in the way of synaptic activity. We also learned that zinc and copper are necessary to drive the aggregation of A-beta in the brain. So based on this, to treat the disease, we come up with the kitchen sink model. This is the ugliest kitchen sink I could find on the internet. I don't know where this sink is. I don't want to live there. But as you can see, the sink is overflowing. So picture the brain overflowing with amyloid. What do? You turn off the spigot, or you unclog the drain. And this is exactly what drug companies have been trying to do, modulate the production of the A-beta or try to clear it out. Now, this gave us our choke points then. Hit production and clearance. Now, this is the good news, is that we have a target. We know what we need to do. The bad news is that the first wave of drugs have not been very good. They've either not been safe, they didn't get into the brain, or if they did get into the brain, they weren't potent enough. So we know that in the second wave, the third wave of drugs, we need to continue on this target. We need to find a better way to do it. How do we do that? And this is where the venture research funding comes in from Cure Alzheimer's Fund to teach us just how to do that. We need to find the rest of the AD genes. I should tell you, those 4 genes only account for 30% of the story. 70% of the genetics remain unknown. And we need to come up with safer and better drugs based on what we're learning from these genes. So we started the Alzheimer's Genome Project with the funding of the Cure Alzheimer's Fund, where we basically screened the entire human genome. We studied 5,000 families with Alzheimer's disease, where there are multiple affected individuals in each family. And this was only possible with Cure Alzheimer's Fund funding. At the time we started this 5 years ago, it was almost impossible to get this type of money from any other source. We got it going early on. We finished ahead of schedule and under budget, actually. And when we published our first genes that came out of this, new Alzheimer's genes, Time Magazine named us a top 10 medical breakthrough of 2008. So here's where we were. Originally, the 4 genes, say, target A-beta. Now, we have over 100 new Alzheimer's gene candidates. I say candidates because we still have to find the defects in these genes. But now we can start looking at these genes and say, what else is going on? What other biological pathways are involved upstream and downstream of that A-beta production? Where do we need to hit? New choke points means new shots on goal for more drug discovery. Well, here's what we learned, and it's in yellow at the bottom of the slide there, is that many of these genes are teaching us that we have to look at lifestyle. We have to look at the environment. That most of the genes that are new that we're finding modulate how your brain responds to insult. What's an insult? Neurovascular event, a stroke, a mini stroke you don't notice, traumatic brain injury due to a fall, maybe a neurotoxin, an infection. As we get older, our brain takes hits. What we've learned from so far, the biggest thing we've learned from the Alzheimer's Genome Project is there's a big box of genes that controls what's called the innate immune system of your brain. Every time your brain gets an insult, whether it be a head bang or a mini stroke or an infection, that innate immune system gets turned on. It's how the brain protects itself. Part of that is the A-beta protein goes up. You're turning up A-beta. Your genetics is the modulator. What we've learned is that when you have an injury or an insult from your life exposure, your genetics determines how you react. If you overreact, too much A-beta. So now this gives us all new ideas. We can think about stopping the insults. Heart healthy, what's good for the heart's good for the brain. Mitigate the risk for stroke. Obesity, diet, exercise, what's good for the heart will be good for the brain because you're going to minimize and mitigate the insults that trigger the A-beta production. If you can't do that, we can go in and try to figure out how does that innate immune system get turned on. Let's come up with drugs that can modulate that response in those who overreact in response to age-related injury. We've also come up with some drugs that can do this, again with Cure Alzheimer's funding, drugs called gamma secretase modulators that I'm working on with my colleague Steve Wagner. My wife, Dora Kovacs, is working on something called ACAT inhibitors. These are candidates for the statins of Alzheimer's because they safely and potently turn down the A-beta production, akin to the way you would use Lipitor to try to control cholesterol levels and bring them down. And I should tell you that A-beta isn't just a bad guy. In the brain, we've come up with roles for A-beta. We were shocked to learn over the last 2 years that A-beta fights bacteria. It's an antimicrobial peptide. It actually is very effective at fighting staph infections, yeast infections. This is probably the reason why when the brain is under attack by some insult, that when the innate immune system gets turned on, A-beta goes up because it's part of that system. So you don't want to completely get rid of it. Just like cholesterol, dial it down to a safe level. Do that with diet, do it with exercise, and if necessary, do it with therapy and drugs. So this is our goal then, to use these genes, what we're learning for early prediction, using genes and an algorithm that will reliably predict risk. We're not there yet. There are companies that say we are. We're not. We can't predict risk for Alzheimer's reliably yet, but we will. But we need all the genes to do it first. Early detection, if you're at risk, by MRI, by biomarkers, and most importantly, use those genes to prevent the disease before it strikes. In doing this, over the next 10 years, our hope, our intention is to eradicate Alzheimer's disease by 2020. So thank you very much for your attention.